Basics of Medical Biochemistry. First edition 2023. Chapter 16. Metabolism of Purines and Pyrimidines, Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tao, M.Philosophy, Ph.D., Dip M.E. Head of Biochemistry Department-Fazaia Ruth Pfau Medical College Air University, PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 16. Metabolism of Purines and Pyrimidines. 16.1 Introduction. Purine and pyrimidine are nitrogen-containing heterocyclic, aromatic compounds. Purines consist of a 6-membered and a 5-membered nitrogen-containing ring, fused together. Types of purines. Adenine, 6-aminopurine. Guanine, 2-amino-6-oxypurine. Hypoxanthine, 6-oxypurine. Xanthine, 2,6-dioxypurine. 16.2 Synthesis of purines. It takes place in the cytoplasm of most cells of the body, mainly in the liver, small intestine, and thymus. Its main purpose is to provide purines, A and G, for energy metabolism and DNA and RNA synthesis. There is no special dietary requirement for the synthesis of purines in the body. Body tissues can synthesize purines from the components of the usual diet. Since purines are not synthesized as such but rather as nucleotides. There are 2 mechanisms for the synthesis of purine, 1 de novo synthesis and 2 salvage pathways. Figure 16.1 Pathways of synthesis of purine. 16.2.1 De novo synthesis of purine nucleotides. Building the bases from simple building blocks, it includes a series of complex reactions. The first purine synthesized is inosine monophosphate, IMP. It is regarded as a parent purine nucleotide and serves as a precursor to AMP and GMP. This mechanism of IMP synthesis requires the following substances. I5-phosphorobosyl-1-pyrophosphate, PRPP. It is derived from the metabolism of ribose by the enzyme phosphorobosyl pyrophosphate, PRPP, synthetase. Figure 16.2 Formation of 5-phosphorobosyl-1-pyrophosphate, PRPP. 2-glutamine, a type of amino acid in amide form. 3-glycine, non-essential amino acid. 4-aspartic acid, non-essential amino acid. V. Active form of tetrahydrofolate, N-formyl tetrahydrofolate. 6-carbon dioxide, obtained from oxidation of foodstuff. 7-ADP, as a source of energy. Figure 16.3 Sources of C and N of purine. Steps of de novo synthesis of purine nucleotides. 1. Formation of 5-phosphorobosylamine. It is formed as a result of the reaction between PRPP and glutamine by the enzyme glutamine PRPP aminotransferase. The AMP and GMP are inhibitors while PRPP acts as an activator for this enzyme. 5-phosphorobosyl-1-pyrophosphate plus glutamine plus H2O. 5-phosphorobosylamine plus glutamate plus PPI. 2. Formation of glycinamide riboside. GAR. The 5-phosphorobosylamine reacts with glycine by the enzyme synthetase to form glycinamide riboside. 1 ADP is consumed in this step. 5-phosphorobosylamine plus glycine plus ADP glycinamide riboside, GAR, plus ADP plus pi. 3. Formation of N-formyl glycinamide riboside, FGAR. The formylation of the free amino group of GAR takes place in this step by the enzyme formyl transferase. The cofactor of the enzyme is N10-formyl tetrahydrofolate. Glycinamide riboside, GAR, plus N10-formyl tetrahydrofolate N-formyl glycinamide riboside, FGAR, plus tetrahydrofolate. 4. Formation of N-formyl glycinamidine riboside, FGAM. The amide amino group of a second glutamine is transferred to FGAR to form N-formyl glycinamidine riboside, FGAM, by the enzyme FGAM synthetase. 1-ADP is also consumed in this step. N-formylglycinamide ribitide, FGAR, plus glutamine plus ADP plus H2O. N-formylglycinamidine ribitide, FGAM, plus glutamate plus ADP plus pi. 5. Formation of 5-aminoimidazole ribitide, AIR. In this step, formation of 5-aminoimidazole ribitide takes place by the closing of the imidazole ring with the utilization of 1-ADP. The enzyme for this step is AIR synthetase or FGAM cyclase. N-formylglycinamidine ribitide, FGAM, plus ADP 5-aminoimidazole ribitide, AIR, plus ADP plus pi. 6. Formation of carboxyaminoimidazole ribitide, CAIR. In this step, C6 is introduced as HCO3 to the molecule of AIR to form carboxyaminoimidazole ribitide by the enzyme AIR carboxylase. 5-aminoimidazole ribitide, AIR, plus CO2 carboxyaminoimidazole ribitide, CAIR. 7. Formation of 5-aminoimidazole 4-N-succinylocarboxamide, ribitide, SAICAR. It is an amide condensation reaction in which N1 is acquired from aspartate by the enzyme SAICAR synthetase. The reaction is driven by the hydrolysis of ADP. Carboxyaminoimidazole ribitide, CAIR, plus aspartate plus ADP 5-aminoimidazole 4-N-succinylocarboxamide, ribitide, SAICAR, plus ADP plus pi. 8. Formation of 5-aminoimidazole 4-carboxamide ribitide, ICAR. In this step, 5-aminoimidazole 4-N-succinylocarboxamide, ribitide, SAICAR, is cleaved by adenylosuccinate lyase to form 5-aminoimidazole 4-carboxamide ribitide, ICAR, with the liberation of fumarate. 5-aminoimidazole 4-N-succinylocarboxamide, ribitide, SAICAR, 5-aminoimidazole 4-carboxamide ribitide, ICAR, plus fumarate. 9. Formation of 5-formaminoimidazole 4-carboxamide ribitide, FAICAR. It is another formylation reaction catalyzed by ICAR transformylase to form 5-formaminoimidazole 4-carboxamide ribitide, FAICAR, from 5-aminoimidazole 4-carboxamide ribitide, ICAR. 5-aminoimidazole 4-carboxamide ribitide, ICAR, plus N10-formyl tetrahydrofolate 5-formaminoimidazole 4-carboxamide ribitide, FAICAR, plus tetrahydrofolate. 10. Formation of inosine monophosphate, IMP. In this step, cyclization or ring closure of FAICAR takes place to form IMP. Water is eliminated by the enzyme cyclohydrolase. 5-formaminoimidazole 4-carboxamide ribitide, FAICAR, inosine monophosphate, IMP, plus H2O. Once formed, IMP is rapidly converted to AMP and GMP, as it does not accumulate in cells. Figure 16.4 De novo synthesis of IMP, AMP, adenosine monophosphate, ADP, adenosine diphosphate, GMP, guanosine monophosphate, PRPP, 5-phosphoribosyl 1-pyrophosphate, Pi, inorganic phosphate, PPI, pyrophosphate. The synthesis of AMP and GMP from IMP. IMP represents a branch point for purine biosynthesis because it can be converted into either AMP or GMP through two distinct pathways. I. Formation of AMP from IMP. The pathway leading to AMP formation requires energy in the form of GTP. The utilization of GTP in the pathway to AMP synthesis allows the cell to control the proportions of AMP and GMP to near equivalence. The accumulation of excess GTP leads to enhanced AMP synthesis from IMP. AMP is made from IMP in two steps. The first step converts IMP to adenylosuccinate by reacting with aspartate in the presence of GTP. Aspartate acts as a source of nitrogen to synthesize AMP. The enzyme is adenylosuccinate synthetase. IMP plus aspartate plus GTP adenylosuccinate plus GDP plus pi. The second step is catalyzed by adenylosuccinase produces AMP along with the release of fumarate. Adenylosuccinate AMP plus fumarate. 2. Formation of GMP from AMP. The pathway that leads to GMP formation requires energy in the form of ADP. The accumulation of excess ADP leads to accelerates the synthesis of GMP over that of AMP. The formation of GMP from AMP also occurs in two steps. In the first step, xanthosine monophosphate, XMP, is produced by the enzyme IMP dehydrogenase. AMP plus H2O plus NAD plus XMP plus NADH plus H plus. The second step, catalyzed by GMP synthetase, is a glutamine-dependent aminotransferase reaction that replaces the oxygen on C2 with an amino group to yield 2-amino,6-oxypurine nucleoside monophosphate i.e., GMP. XMP plus glutamine plus H2O plus ADP GMP plus glutamate plus AMP plus PPI. GMP synthesis from IMP requires 2 additional ADP equivalents for a total of 9 ATPs. Mycophenolic acid is an irreversible inhibitor of IMP dehydrogenase. It is used as an immunosuppressant drug to prevent graft rejection. Figure 16.5 Synthesis of AMP and GMP from IMP. AMP is also known as adenylate, GMP is also known as guanylate, NAD, nicotinamide adenine dinucleotide, GDP, guanosine diphosphate, GTP, guanosine triphosphate, AMP, adenosine monophosphate, Pi, inorganic phosphate, PPI, pyrophosphate. 16.2.2 Regulation of de novo synthesis. The significance of regulation is concerned with meeting the needs of the body, without wasting. The most important regulatory step for this mechanism is the first step, which is catalyzed by the enzyme glutamine PRPP aminotransferase. This enzyme is inhibited by AMP and GMP while activated by PRPP. The second regulatory step is the synthesis of PRPP from ribose 5-phosphate by the enzyme phosphoribosylpyrophosphate pyrophosphate, PRPP, synthetase. This enzyme is allosterically inhibited by ADP. The third regulation is found in the formation of XMP from IMP. This step is inhibited by an excess of GMP. Figure 16.6 The regulatory circuit controlling purine biosynthesis. Allosteric regulation occurs in the first 2 steps, whereas AMP and GMP are competitive inhibitors in the 2 branches at right. 16.2.3 Salvage pathways for the synthesis of purine nucleotides. Purine bases created by the degradation of RNA or DNA, an intermediate of purine synthesis, can be directly converted to the corresponding nucleotides by salvage pathway. The significance of the salvage pathway includes saving of fuel, because of the high energy cost for de novo synthesis of nitrogen bases. Some tissues and organs such as the brain and bone marrow are only capable of synthesizing nucleotides by salvage pathway. 2 phosphoribosyl transferases involved in this recycling mechanism are adenine phosphoribosyl transferase, APRT, for adenine, and hypoxanthine guanine phosphoribosyl transferase, HGPRT, for guanine or hypoxanthine. Salvage pathways collect hypoxanthine and guanine and recombine them with PRPP to form nucleotides in the HGPRT reaction. Formation of AMP from adenine. Adenine reacts with PRPP to form AMP by the enzyme adenine phosphoribosyl transferase, APRT. Adenine plus PRPP AMP plus PPI. Formation of GMP from guanine. PRPP reacts with guanine to form GMP by the enzyme hypoxanthine guanine phosphoribosyl transferase, HGPRT. Guanine plus PRPP GMP plus PPI. Formation of IMP from hypoxanthine. PRPP reacts with hypoxanthine to form IMP by the enzyme hypoxanthine guanine phosphoribosyl transferase, HGPRT. Hypoxanthine plus PRPP IMP plus PPI. 16.3 Catabolism of purine nucleotides. The catabolic process starts from purine-containing nucleosides, i.e., adenosine and guanosine. These nucleosides are derived from respective nucleotides which are released from the breakdown of RNA and DNA. Adenosine is obtained from AMP by the enzyme 5-nucleotidase. AMP plus H2O adenosine plus pi. Similarly, guanosine is obtained from GMP by the same enzyme, i.e., 5-nucleotidase. GMP plus H2O guanosine plus pi. 16.3.1 Catabolism of adenosine. It is first converted to inosine by adenosine deaminase, a nucleoside deaminase. Adenosine plus H2O inosine plus NH3. Inosine has purine called hypoxanthine. Inosine is also derived from AMP through the following reactions. H2ONH3H2O AMP inosine. AMP deaminase 5 nucleotidases. The ribose of inosine is then removed from inosine by purine nucleoside phosphorylase to form hypoxanthine. Inosine plus hypoxanthine plus ribose 1-phosphate. Hypoxanthine is oxidized to xanthine and then finally to uric acid by xanthine oxidase. H2O plus O2 H2O2 H2O plus O2 H2O2. Hypoxanthine xanthine uric acid. Xanthine oxidase 16.3.2 Catabolism of guanosine. It is first converted to guanine by the removal of its ribose by the enzyme purine nucleoside phosphorylase. Guanosine plus guanine plus ribose 1-phosphate. Guanosine is deaminate to xanthine by guanase. Guanine plus H2O xanthine plus NH3. Xanthine is oxidized to uric acid by xanthine oxidase. The end product of purine catabolism in man is uric acid. Other mammals have the enzyme urate oxidase, uricase, and excrete the more soluble allantoin as the end product. Humans do not have this enzyme, so urate is the end product for us. Uric acid is formed primarily in the liver and excreted by the kidney into the urine. The normal range of serum uric acid level in males is 3.5-7 mg/dl and in females is 3-6.5 mg/dl. The uric acid pool in males is about 1,200 mg and 600 mg in females. The total excretion of uric acid is in the range of 500 to 600 mg/24 hours. About 80% of the uric acid formed in the body is excreted in the urine and the remaining part is probably excreted in the bile. The high acidity of urine, about 5.8 pH, prevents sodium urate formation, which is much more water-soluble than uric acid. Limited solubility is not a problem under normal circumstances unless the urine is very acidic or has high Ca++. Urate salts precipitate with calcium salts and can form stones in the kidney or urinary bladder. Besides uric acid, some other purine derivatives are also present in urine such as adenine, guanine, hypoxanthine, xanthine, and methylguanine. 16.3.3 Hyperuricemia and gout. Hyperuricemia is the condition of increased, above 7.0 mg/dl, levels of uric acid as urates in the blood, accompanied by increased excretion of uric acid in the urine. It results from the increased activity of the phosphorobosyl pyrophosphate, PRPP, synthetase enzyme. This leads to uric acid overproduction and excretion. Gout occurs from the accumulation of uric acid crystals in the extremities. This results in the deposition of sodium urate in the form of monosodium urate needle-like crystals on the joint linings and in soft tissues around it, especially those of the big toe, skin, kidney, and other tissues. The precipitated urates are called tophi. It results in inflammatory reactions in the joints, i.e., acute gouty arthritis that progresses to a chronic form. Uric acid and urate precipitate in the kidney and urinary tract, which may result in stone formation. The chances of urinary stone formation are 1,000 times higher in patients with gout than in normal people. Tophaceous gout is characterized by chronic accumulation of monosodium urate, MSU, crystals as tophi. The hereditary fact of gout is reflected by the 25% involvement of family members of the gouty patient. There is an increased risk of cardiac problems due to secondary hyperlipidemia, which is frequently associated with hyperuricemia. Management of gout includes both medication and dietary care. One of the drugs used is allopurinol, a competitive inhibitor of the enzyme xanthine oxidase. It decreases the formation of uric acid and increases the excretion of xanthine. Xanthine is more water-soluble and thus chances of urinary stone formation are minimized. Patients are advised to avoid purine-rich foods like all meats, seafood, beans, peas, lentils, spinach, cauliflower, mushrooms, yeast, beer and other alcoholic beverages. They are encouraged to take low purine foods like cereal products, corn flakes, pasta, white bread, flour, sugar, gelatin, fruits, lettuce, tomatoes, other green leafy vegetables, nuts, and peanuts. Primary causes of hyperuricemia. 1. Metabolic causes. These are due to inherited autosomal or X-linked recessive metabolic defects, congenital enzyme deficiencies, characterized by an increased rate of purine synthesis and/or decreased rate of salvaging purine from the breakdown pathway, leading to overproduction of uric acid. There is increased utilization of glycine to form purines due to inherent metabolic defects. Examples include Lesch-Nyhan syndrome and von Gierk's disease. Lesch-Nyhan syndrome, it is caused by a severe deficiency in HGPRT activity. There is a failure to salvage hypoxanthine or guanine. Symptoms are gouty arthritis due to uric acid accumulation and severe neurological malfunctions including involuntary movements, mental retardation, aggressiveness, and self-mutilation. It is a sex-linked trait occurring mostly in males. Lack of HGPRT activity in Lesch-Nyhan syndrome causes a buildup of PRPP, which activates the synthesis of purine nucleotides. Hypoxanthine guanine. Phosphoribosyltransferase. Guanine plus PRPP x guanylate plus PPI. Hypoxanthine plus PRPP x inosinoate plus PPI. Von Gierk's disease, type I glycogen storage disease. It is the metabolic disorder of glycogen utilization. The absence of glucose 6-phosphatase shuttles glucose 6-phosphate to HMP shunt with excessive production of ribose, a precursor for purine synthesis. 2. Primary renal gout. It is a rare condition and is due to an inherited defect in the kidney leading to decreased secretion of urates by the renal tubules. Table 16.1 Secondary causes of hyperuricemia. I. Increased catabolism and turnover of purine. 2. Decreased renal clearance of uric acid. 1. Myeloproliferative disorders. 2. Lymphoproliferative disorders. 3. Carcinoma and sarcoma. 4. Chronic hemolytic anemias. 5. Cytotoxic drugs. 6. Psoriasis. 1. Intrinsic kidney disease. 2. Functional impairment of tubular transport. I. Drug-induced, thiazide, low dose. Aspirin. 2. Hyperlactic acidemia. Lactic acidosis, alcoholism. 3. Hyperketoacidemia. Diabetic ketoacidosis, starvation. 4. Diabetes insipidus. Xanthinuria is a rare genetic disorder causing the accumulation of xanthine. It is caused by a deficiency of the enzyme xanthine oxidase. It may be due to prolonged and excessive use of the drug allopurinol. There is increased excretion of xanthine in urine. Serum uric acid is low. In severe cases, xanthine calculi may be formed. As xanthine stones are radio-opaque, so missed by routine radiography. Exercise questions. 4 MCQ 6 to 10. See Part 2 of this chapter. MCQ 1. A 33-year-old man consulted a doctor for complaints of painful swelling of the first metatarsophalangeal joint of his left foot. Lab reports also showed raised serum uric acid levels. Which of the following nitrogen bases can produce a substance that was found raised in this patient? Adenine, choline, cytosine, thymine, uracil. MCQ 2. A 42-year-old man suffering from cancer underwent radiation therapy and developed severe pain in his right big toe. Laboratory analysis showed elevated serum uric acid level and urate crystals in his urine. This patient's pain is caused by the overproduction of the end product of which of the following metabolic pathways? De novo purine biosynthesis. De novo pyrimidine biosynthesis. Purine degradation. Purine salvage. Pyrimidine degradation. MCQ 3. A 6-year-old boy presented with complaints of aggressiveness and self-mutilation. Physical examination revealed delayed motor development, exaggerated reflexes, and spasticity. Lesch-Nyhan syndrome is strongly suspected in this boy. Which of the following compounds is expected to be elevated in the urine of this patient? Adenosine, guanine, hypoxanthine, uric acid, xanthine. MCQ 4. The condition of orotic aciduria due to defects in de novo pyrimidine biosynthesis can be treated by the administration of which of the following compounds? Cytosine, deoxycytidine, thymidine, uridine, xanthine. MCQ 5. A 7-year-old boy presented with swollen tender joints. History revealed a tendency to bite his lower lip and chew his fingertips. These symptoms and lab results are indicative of a deficiency of the enzyme HGPRT. Which of the following compounds is expected to be elevated in the serum of this patient? Bilirubin, cytosine, glycine, orotate, xanthine. End of Part 1 of 2 of Chapter 16. Thank you.